Good afternoon, everyone. Hope you can hear me. So there is going to be a slight delay, about 5 minutes for this particular session, which is focusing on the bioinformatics, the session 3A. There is also another session going on parallel to this session called 3B, which is focusing on applications of IT. So please sorry for the inconvenience, and we'll start the session at 3:55. Good afternoon again, everyone. First of all, I would like to introduce our session chair, Dr. Champi Tusangi Vanige. Let me give a brief introduction about her first. She received her PhD degree in computational systems biology from Lincoln University of New Zealand in 2014. She also received her BSc Special and Master of Philosophy degrees in Computer Vision from University of Colombo in 2007 and 2010 respectively. And she is currently working at the Department of Computer Science of University of Ruhuna as a senior lecturer. Her research interests are bioinformatics, systems biology, modeling and simulation, and machine learning. So with that, I would like to hand the session over to you, Dr. Champi.
Okay, I think you all can hear me, isn't it?
Yes, we can hear you.
Very good afternoon, and I warmly welcome you to this bioinformatics session. So there are four interesting researchers we have to discuss in this session. So the first research work is on paper ID 73, and the title is Genomic Data Analyzing Workflow for Single Nucleotide Polymorphisms in Human Nervous System Cancers. So this is going to be presented by SPBM Senadira. I think we can play his video now.
Hello, I'm DATASET and my supervisor is Dr. A.R. Veerasinghe. Our research topic is genomic data analysis workflow for single nucleotide polymorphisms in human nervous system cancers. Overview of our presentation starts from introduction, problem statement, objectives, methodology, results, discussion, and conclusion. Central dogma is a biological concept which explains how protein is synthesized. DNA will encode RNA, and from those RNA, protein will be encoded. If the mutation occurred in DNA level, it will represent in the protein level, which will lead protein structure can be changed or damaged. So this concept is very vital in cancer data analysis also. Our research is focused on identifying these critical mutations and also developing a workflow of mutation analysis, since there is no proper integrated workflow for mutation analysis. Objectives of our research are as follows. We are going to build a workflow which can be used for human nervous system cancer mutations, and also try to identify significant mutation positions in human nervous system cancer datasets. And also we are going to generate an information-rich dataset for analyzing human nervous system cancers. This is the methodology that we are going to use in this research. We will start from the data purification, and we will annotate data in genomic level, and then in the protein sequence level we also try to annotate these mutations, and then protein structure level data annotation will be done for mutation that we have identified from the dataset, and then the data visualization. This is the dataset that we have used for this research. It has 4 peripheral nervous system cancer types and also 11 central nervous system data sets. We have used SNP mutations, that means single nucleotide polymorphisms, that are the main type appeared in the dataset. However, we have eliminated the large deletions, insertions in this analysis. We have used two methods to annotate genomic level data. Direct annotation, we call it in method 1, and the indirect annotation method is the method 2, which we have converted the old genomic position to new genomic positions and then converted to the protein position that we are interested in. This is because in the method 1 we have identified three main issues. Some protein positions do not provide genomic positions since there can be tools errors. And there are protein positions mapped with different chromosome genomic positions such as P62805, which will encode to chromosome 1, 6 and 12. This is because they are from histone proteins. And there are some positions which are represented in protein level SNPs. However, in the genomic level, they are not a single nucleotide polymorphism, rather than they are large deletion or insertions. So we have to use the second method, that we have to use the genomic level conversion in order to map the protein position. So we have compared two different tools and two different databases for mapping these genomic positions. So here we have understood that uniprot mapping is the most significant mapping compared to the web mapping tool. So which use the another database called Ensemble. So here we use the uniprot mapping for protein identification. So later for the protein level mapping, we have to consider the PDB, BISQ, and Toastmaster group tools for the analysis. However, these tools may have different advantages and disadvantages. Some of them could not process batch processing, and some of them could not identify the exact protein position. So with that, we have we have merged some tools in order to perform our task. So we have used the PDB genomic mapper and the BISQ tool for our analysis. So these are the findings in our research. Genomic positions in highest genomic alteration would appear in the chromosome 2, and it has 639 patients are mutated in that position. And then for the protein level mapping, we have used the isoforms and also the proteins differently. And then we have identified what are the highly mutated isoforms in our data set. And these are from main cancer genes such as IDH1 and TP53. So we have mapped these for the amino acid frequency also. R to H is the highest mutation pattern we have identified in our data set, and this is also a significant biasness identified in biological laboratory experience also. So this finding explain that there is high chance of getting mutated R to H. So the conclusion of our research is all mutations should be converted to the latest genomic build before we start the analysis, and merging web enrichment data with uniprot data rescue tools would provide more insight about cancer mutations, because it gives more information about the mutation positions, and we have generated reusable effective workflow for genomic data enrichment and data mining, and we have generated a data set for human nervous system tumor SMP mutations, and it has all genomic and protein level information. In future, this data set needs to be enhanced by adding more tumor types such as lung cancers and other types of cancer, subtypes of cancer. Thank you.
Okay, thank you very much. So now it's open for questions. I have a single question to ask from you. So actually, how did you validate your workflow?
Uh, I have validated the workflow by using the other tools, their performance. So we have in the genomic level, we have mapped with the two databases, and from that we have come across that our the database based on the database that we are using, it can give more results than we have used two databases, Ensemble and Uniprot. So most of the tools use a single database mapping or annotation. So in this workflow, we are using both databases for this annotation. So it will improve the results around because it's kind of merging the results. So that is one map validation. And for the protein mapping validations, we use PDB mapper and Uniprot web portal, because since they have different as an example, in Uniprot you can't get the exact position for the protein. They are giving the starting position and the end position. So when we use the API and implement it with the R, they will only they will give the exact position in that scenario only. So we have compare like that, but we have to map. I mean, we have validate with the user friendliness with other clinicians and so on. We didn't do it yet.
Okay, so validation is really important when you are introducing the workflow. Is it generalizable for other problems as well?
Yes, it can be generalizable for other cancer types. However, I'm not sure with the other mutation mapping, since for other species or some like mapping plants or something else, it will be different, because I'm using particularly for human mutations.
Did you come
So
up with
yeah,
a percentage of accuracy or something like that?
sorry.
Did you come up with a percentage of accuracy or something like that?
Yeah,
Which,
in the genomic mapping, we get around 90% mapping we could achieve. However, there are some errors in the databases, the version controls. So we contact the databases and so on, because they also said it's not 100% validated, because with the genome versions, it will change sometimes. In like example, like in the chromosome, they have mapped for the 6, 12 and so on. So there are some conflicts in the biological context, so we couldn't achieve 100%. However, it has to be updated with the databases versions. The workflow should be always updated with the versions.
okay, so it's open for the questions from the audience. Okay, so I think it's time to move to the second presentation. Thank you very much, Senadira. I don't know your first name, sorry. So the second presentation is for the paper ID 61, which is the paper title is Dimensionality Reduction for Cluster Identification in Metagenomics using Autoencoders. So this is presented by Kalana Vijay Gunaratna, who is a computer science and engineering graduate at University of Moratuwa. Currently, he is researching in the field of bioinformatics for his final year project. So Kalana, so it's for you to present your work.
Excuse me, madam. We are waiting for few evaluators to join for this session. So we will take a few minutes until they join from the other session.
Sorry for the inconvenience, everyone. It seems like we have to wait some more time because I think the other members of the panel are in a Q&A session, ED session 3B. So we might have to wait a little bit more.
sorry for the inconvenience caused. We will now start the presentation of the second presenter.
Welcome, ladies and gentlemen. My name is Kalani Vijayagunratna, and the topic of our paper and the presentation today is dimensionality reduction for cluster identification in metagenomics using autoencoders. Genomics is a branch of biology studying the genome structure, function, evolution, mapping, and editing. Metagenomics, on the other hand, analyzes similar but not identical microorganisms that are derived from environmental samples or natural habitats. For example, from a soil sample or the gut of a blue whale. While in genomics, the goal is to determine the sequence of the genome and the genes function in an organism, metagenomics focuses on the function of the genes found in a community. Most problems in the domain of genomics pertaining to the field of bioinformatics can be grouped as pattern recognition or anomaly detection problems of genomic sequences extracted using various technologies. Visualizing of the metagenomic sequences understandably have an even greater importance because at the time of acquisition, the researchers have no idea how many species are there in the sample or which of the metagenomic reads belong to which species. Genomics is constantly plagued with the problem which is generally known as the curse of dimensionality. With the latest frequencing technologies, though there is ample data, this data is hardly visualizable or analyzable due to their high dimensionality. Currently, most commonly used dimensionality reduction approaches are PCA and TSNE, but these techniques have their limitations. In addition to having reduced scalability, accuracy, and capabilities, I will go on to show later in my presentation why some of these techniques are at a unique disadvantage in the field of metagenomics. The technique of using autoencoders for dimensionality reduction in metagenomics that I present to you today will prove to overcome these limitations and also better suit the unique requirements of metagenomic visualizations. Firstly, it is important to understand the various techniques that can be used for dimensionality reduction in general. Studying the works of van der Marten on TSNE, which is an improvement to stochastic neighbor embedding, SNE, by Hinton and Royce, and the works of Wold, Esbensen, and Ghilardi on PCA helped kickstart our research. Gisbrecht in his works presents a comprehensive comparison on the performance of PCA and TSNE in the field of metagenomics and points out their pros and cons. Notably, PCA's limitation to being a linear model, but preservation of the global structure, which is ignored in TSNE, although it is an unlinear technique. PCA also shows little data preservation, while TSNE is inefficient in large values of data. The use of autoencoders in genomics is an under-researched topic. As a matter of fact, the improvements of neural network are yet to catch on. Ursulin's deep count autoencoders have been used for denoising single-cell RNA sequence datasets, but no literature backing any research on the use of autoencoders for dimensionality reduction in the field of genomics was found. Understanding the concept of oligonucleotide frequencies or chemofrequencies is vital to understanding our research. The frequency at which oligonucleotides or chemos occur in genomic sequences is unique across the microorganisms. They are species-specific signatures. It is these high-dimensional vectors that represent a particular read in a metagenomic sample, and they act as the input to any of our dimensionality reduction techniques. This slide demonstrates a simple calculation of 3-mer or trinucleotide frequency vectors. A simple string of DNA written using the 4 letters A, C, G, and T is shown. First, we calculate all the possible combinations. There is a total of 64 possible combinations of 3 letters. 3 letters because it's a 3-mer. Then we count the frequency of occurrence for all these combinations. This resulting vector is a 64-dimensional vector. But as is common practice, reverse complementary chemos can be added as pairs. This will result in 32-dimensional vector, which is the vector which needs to be visualized. These 32-dimensional vectors that we spoke of cannot be visualized for a human to interpret because of their high dimensionality. This is where dimensionality reduction comes into play. Traditionally, TSNE or PCA or a combination of both is used to reduce these 32-dimensional vectors into 3 or 2-dimensional vectors. And what we shall be doing is we will be replacing the PCA and TSNE using autoencoders for dimensionality reduction. What you see here is a simple generic architectural diagram of an autoencoder. An autoencoder basically consists of an encoder and a decoder, and in the middle you have the bottleneck layer. And in our case, the bottleneck layer was always a two-node layer, so that we can obtain a two-dimensional vector from the bottleneck layer. And there were multiple hidden layers between the input layer and the bottleneck layer, which is not shown here because it's a simplified diagram of an autoencoder. Our dataset was based on Gisbert's comparison of TSNE and PCA. We recreated his results using TSNE and PCA, and on top of that, we tested the performance of autoencoders. What you see on the right are the microorganisms that we use in our dataset. And just like Gisbert, we had to create a synthetic metagenomic dataset because in order to compare the techniques, we need to know the ground truth labels of the reads. We obtained the genomes from NCBI and created a metagenomic dataset of reads with an average length of 5,000 base pairs and standard deviation of 1,000 base pairs. Now let's take a look at the pipeline of our experiment. The data preparation step is what we just went through with the creation of synthetic metagenomic dataset, counting of the k-mers, forming the k-mer frequency vectors, and also we normalize these k-mer frequency vectors because neural networks tend to perform better on normalized data. And then we come to the training phase where the autoencoder is trained such that the input vector is reproduced at the output layer of the autoencoder. So we train it until the loss in the bottleneck layer is minimal. And then we come to the dimensionality reduction phase, where here again we feed the autoencoder with the same input vectors, but we do not let the autoencoder finish its process. We intercept it in the middle, and from the bottleneck layer we take out the two-dimensional vectors of those high-dimensional frequency vectors that we fed the autoencoder. And these two-dimensional vectors that we obtain from the bottleneck layer are then used for the 2D visualizations in scatter plots. So now since we are doing a comparison between these techniques, we need to be able to put a rating or a number on the clusters formed. So in order to evaluate the clusters, we use dbScan to detect the clusters. And then the evaluation is carried on the dbScan clusters that has been, the clusters that dbScan identifies in these scatter plots that were produced from the autoencoders, PCA and TSNE's dimensionality reductions. When evaluating the goodness of dimensionality reduction, there are two aspects that need to be evaluated. One is the retention of information in during the reduction of dimensionality, and the other is the formation of clusters. Now, there could be an instance where clusters are very clearly separated, but if you consider the ground truth labels, the points in the same cluster are not actually related in their higher dimensions. That is one case. And then the other case is where the clusters don't seem that good, the clusters don't seem that clear, but still the information retention is very good. So in order to get a clear understanding of which dimension reduction technique is better, we need to evaluate them on two aspects, which are the using the ground truth labels, and the other one is the goodness of clusters. So the validity measure is a supervised technique that uses the ground truth labels. The validity measure is the weighted harmonic mean of homogeneity and completeness, while the Sill-out coefficient measures how clear the clusters are and how close the points in the clusters are compared to how distant the points of different clusters are. And the Sill-out coefficient does not require prior ground truth labels. With that, we can move on to the results of our experiment. This table shows the results of the tetranucleotides, how the tetranucleotides performed on our autoencoder, and the autoencoder we used is a autoencoder with 5 layers, 136 input layer with a 64-dimensional hidden layer and a 2-dimensional bottleneck layer. So as you can see, the V measure, which is the measure that measures how measures the retention of information during the reduction of dimensionality, is considerably higher than TSNE's, and the sill-out coefficient is slightly lower than TSN is. I will show you the reason as to why this is. And as you can see, PCA has performed well below TSNE as well. So the comparison is really between autoencoders and TSNE. This is the performance of tetranucleotides. If you move on, here you can see the performance of trinucleotides, where the autoencoder used is a 5-layer autoencoder with the 32-dimensional input layer and a 16-node hidden layer and a 2-dimensional bottleneck layer. Again, the V measure is considerably higher than the TSNE's performance, while the sill-out coefficient is lower than the TSNE's sill-out coefficient. And PCA again, as even Gisbest suggests in his papers, performs considerably lower than TSNE. One of the most important observations we made during this experiment is the improved V measure in autoencoders as opposed to the V measure of PCA and TSNE. This improved V measure is a reflection of how well data is preserved during the reduction of dimensionality. That is, how little information is lost during this process in autoencoders than in TSNE during dimensionality reduction. And two other points are taxonomic data preservation in autoencoders and global structure retention in autoencoders than in TSNE. Both of these points are related, and I will go on to elaborate in the next slide. What you see here on this slide is the autoencoder's performance on tetranucleotide frequencies. The 2D scatter plot you see on the left hand side is colored using the ground truth labels, as in this. The yellow color dots belong to the Clostridium phytomenetas, and the green color dots here belong to Clostridium vijayanki, and the other colors belong to other microorganisms. But what you see on the right hand side is the same plot, but the clustering has been done using DBSCAN, and then these are the clusters that DBSCAN has identified as separate clusters. As you can see, it has identified these two, the Clostridium 2 Clostridium bacteria, as two different microorganisms, which is accurate. And also, as you can notice here, the black cluster is separated into two different clusters by DBSCAN, which is not ideal. But what is worthy of notice is that these two bacteria, which are from the same genus, are actually in close proximity. They are closely related to each other, and they are in close proximity even in this 2D scatter plot, even in this reduced dimensionality scatter plots. Now, when you take into consideration the performance of TSNE on the same data set, what you will notice is that the Clostridium bacteria is further away from the other Clostridium bacteria, which is the Clostridium vijayanki, as it is further away from this black color microorganism, which is not even in the same genus as these two. But ideally, these two should be close to each other so that it does not, it could be misleading to a researcher to see these two further apart from each other, just like these two are apart from each other. But in fact, these two are in the same genus, so they should be, that information is lost when reducing the dimensions using TSNE, while it is not the case when doing so with autoencoders. And also, if you look closely, you can see that DBSCAN has found several smaller clusters within these larger clusters, which it has found 15 clusters, which is not the real case. It is actually 8. Ideally, should have been 8 clusters, but it is these smaller clusters and these compact smaller clusters that that lead to a better sill-out coefficient in TSNE rather than in autoencoders. But this is not an ideal case because this is not important, and these small clusters are actually wrong, and they are not actually real clusters that should have been there in the first place. And here again, this is the performance of autoencoders in trinucleotide frequencies. Here again, these two are in close proximity, but as you see on the right hand side, DBSCAN has not managed to clearly separate those two as two different clusters. And this is the performance of PCA on the tetranucleotides. As you can see, it is well below par, and it performs worse than both TSNE and autoencoders. These are some of the follow-up work that we intend to make on the findings from the autoencoder. Actually, in fact, the tool that we intend to make is already complete, and the paper we wrote on that tool was accepted by the IEEE conference on bioinformatics and biomedicine this year. And features like coupling autoencoders with BLAST is already incorporated in that tool. In conclusion, we believe that these results demand a place for autoencoders in the field of bioinformatics. Thank you very much and have a great day.
Thank you, Kalana. So, Kalana, can you please turn on your camera so that we can see you and discuss? So, I have one question. So, what would happen if you increase the number of genomes or number of species?
That is not a problem with autoencoders, ma'am. And we did try it with TSNE. TSNE tends to perform worse when you increase the number of species or the number of data points, in fact. But with autoencoder, that is not a problem. With neural networks, the more the data, the better it performs. But slightly, there can be longer training time.
So, you mentioned that you validated with real biological data, isn't it?
Yes, the data set we created was synthetic, but what we created the data set from was actual biological data, just like Gisbert did in his comparison. Because we needed the ground truth labels. I mean, without the ground truth labels, if we just use the meta-genomic data set, we wouldn't know which species they belong to. So we wouldn't be able to get a validity measure to compare the two.
How many experiments did you carry out? Only one data set?
No, we carried out multiple experiments with other combinations of data from Gisbert's dataset.
Now it's the others to ask questions.
What is the novelty of your work? Now, in your paper, the 18th reference is a very similar work where you have this visualization, but your paper is different. I see that. It's not only the visualization, but what is the novelty in your paper with respect to your the reference you're given in 18th reference in your paper?
18th is zero inflated factor analysis, right? Is that what you are referring to?
It is also a visualization of
self-organizing map,
by some Chinese people.
18th is E. Pearson and C. Yao Zifa dimensional reduction for zero inflated. Are you referring to the paper?
Self-organizing map, no?
Is the work done in that paper is very similar to this. They are also doing this genome dimensional reduction and visualization. But I can see your paper is different. But I'm asking what is the novelty in this paper?
If you're comparing it to another paper, I would like to know which paper it is, because 18th, as you say, is using ZIFA, that is zero inflated factor analysis.
1818 is using self-organizing map, no? Using self-organizing map.
That's okay. All right. Yeah. So that is using.
I think he's referring to 17 paper.
Is that the single cell RNA sequencing denoising using deep count autoencoders?
Exactly, that's the one. Yeah.
All right. Yeah. So that I referred to that as well earlier. That is using single cell. This is about denoising data. So actually, in our experiment, we did not use noisy data. We could have used noisy data, but this is not the same. This is not about reducing. It's not about dimensional reduction. It has nothing to do with dimensional reduction. What this uses is they are denoising the data. This was actually just this was the only reference we found to autoencoders in metagenomics or in the field of metagenomics about autoencoders. So that is why we use it here. But it has nothing to do with dimensional reduction. It's about denoising the data with autoencoders, deep count autoencoders. And then because single cell RNA sequence data sets usually tend to have a lot of noisy data. So what they do is they denoise it and then they use whatever other methods they have with their next steps, which is not exactly, it does not exactly end with dimensional reduction.
So what is novelty in your work is basically comparing these three different methods, is it?
We propose autoencoders as a dimensional reduction technique, which has not been proposed anywhere. We did not manage to find any other reference to where autoencoders have been used to denoise, reduce dimensionality of genomic data.
So then a few other questions. Then you're using DBScan to cluster, right?
Yes.
So how do you how do you know that DBScan can cluster exactly correctly? So is that algorithm is perfect? Do you know exactly?
Yeah. Now, thank you for the problem. The whether DBScan is the best clustering method to be used is of question. I mean, it's questionable. But that is what even Gisbert says in his paper, that what you can use. There are several arguments to and against. I mean, there can be other techniques that can be used. But why we use DBScan was because DBScan manages to capture clusters with arbitrary shapes, and it can deal with lots of data. I mean, they have not established which is the best clustering technique to be used in genomics. That is not an established fact. It's still up to debate. And there's another method called affinity propagation that could have been used, but it sometimes uses human intervention as well. But as a comparison, if we were to use it, it might feel a bit, it's not very, it does not play up to our integrity.
And one last question, and now you had that small cluster points nearby the clusters. So where do the points come from originally? So are they can't be not, I mean, why do you, from where, which clusters they are coming from? Now in your plot, you get some points which don't clusters, right, nearby. But they must be originating from somewhere, no? So you are saying that they don't belong to any cluster, or what do you mean by that?
Which points are you referring to? All these belongs to some species or some cluster. All these points belong to some microorganism. But sometimes DBScan does not manage to capture all of them into one particular cluster. That is what happens. So I mean, it is not ideal, but it happens in both cases. Even with TSNE, which is the widely used technique, that tends to happen. But I'm not sure whether I'm answering your question properly. I'm not sure about your question. Is it?
No, exactly. Now, in your one of your last slides, there were some points. You said they are outside, right? So they are outside, but they are originating from some clusters, right? So they don't belong to one of your DBScan clusters, but they should belong to one of these clusters, right?
No, DBScan clusters the main clusters, and then it counts others as outliers. So the points you are referring to, I think in the video I showed you, there was a set of points between what is it, the yellow Clostridium phytometas.
How many clusters you expected? 8?
It should be 8.
15.
It should be 8. Yes.
So 15s are appearing.
15 is appearing with TSNE, yes.
So
and with ours we have 9.
Those autoencoders are outliers.
Not exactly outliers, but they have broken those main clusters into smaller categories, smaller clusters. That is what has happened with TSNE. Why that happens is because...
How it happened in your system now?
TSNE is not exactly our system. Autoencoders is our system.
Yeah.
Our solution we are proposing is autoencoders. So with autoencoders, it has happened less if you compare, because TSNE DBScan tends to cluster the TSNE results into 15 groups, 15 clusters, which is wrong. But with ours, it clusters into 9 clusters. DBScan tends to cluster our data into 9 clusters. It should have been ideal if it was 8, but it's not 8, it's 9. With TSNE, it clusters into 15. Why? That is because TSNE has a problem where when it clusters similar points together, and then it randomly pushes away the dissimilar, the points that are not very close to those points. It does not consider relative distances between two clusters. It pushes similar points together and pulls similar points together and pushes dissimilar points away from each other. So that is why this happens, and then they get smaller clusters within these large clusters. That is what DBScan identifies as smaller clusters within those large clusters.
So your one is having purity problem, no? Cluster purity.
No, I wouldn't say it has a purity problem. Why would you say it has a purity problem?
9 because 15. So your accuracy about 80, 89 or 90, no? The cluster accuracy?
Autoencoders?
Yes.
Yes, 93.2 with tetranucleotides and 90.1 with trinucleotides. Yes.
That is because of one extra cluster. So 8 plus 1. You mean
Yeah,
all
yeah.
8 are very good.
I'm sorry?
Out of 9, the 8 are good.
Out of the 9 clusters DB-scan has identified for our output, yes, 8 are pretty good. Yes, 8 are good. You could
So
say
this
that.
is this data experimental data or just generated data? Because
No,
if
these
it is,
are, yeah.
the original data is derived from experimental data or it is generated data. So
These
if it's
are actual.
how do you if it is experimental data, how do you deny the fact that there could be some variant of a species? No,
Yeah, so this data was actually obtained from NCBI. So this is real data. These are actual microorganisms genomes that we have taken, but the production of the synthetic data set is that is synthetically produced, because just as it is, you can't feed the whole genome. You need to break the genomes into small parts like a metagenomic data set. So if you have seen a metagenomic data set, what you get are reads. So reads can be 500, 600, or tend to be even longer than that, 2000, 3000 base pairs long. So that making of those smaller reads that we had to do using a synthetic, we had to make a synthetic data set using those actual genomic data sets. But we used actual real life microorganisms genomes.
So they are the real theoretical genomes that they should have, no?
Yes.
Okay, right. Thank you.
You're welcome.
Okay, thank you very much, Kalana. So let's move to presentation 3. So the paper ID is 112, and the title is Analysis of Expression Data Using Unsupervised Techniques. So this is going to be.
Excuse me, ma'am. Can you hear me? So there's a small announcement to make for the best paper panel. So they have to attend to another Zoom meeting arranged prior by the organizing committee. So that's the announcement. Yeah, thank you.
Now Amali Iresha Pereira would be presenting her work, and Amali Pereira received the BSc Honours degree in Computer Science from Computing in 2019. Currently, she works as assistant lecturer in the Department of Information Systems Engineering at the University of Columbus School of Computing. Her research interests include machine learning, bioinformatics, and image processing. So, Amali, please present your work now.
Hello everyone. I would like to warmly welcome you all to the research analysis of expression data using unsupervised techniques. I'm M.A. Pereira from University of Columbus School of Computing. Before moving deep into this research, I would like to go through some basic topics. In human body, there are different kinds of cells. Cells are the basic unit of all living tissues. Most of these cells have a nucleus inside. In this nucleus, there are 46 chromosomes in human body. 23 came from our mother and 23 came from our father. If we look more closer to these chromosomes, we could see that there are bundled up DNA strands inside it. If we pull this DNA strand and look more closer, we could see that these DNA strands are bundled around the proteins called histones. Again, if we pull this more and look more closer, we could see that there are AT and CG base pairs. These are the base pairs which build these DNA strands. So basically, in human body, there are 3 billion of base pairs. In this DNA, in this genome, we have subsections called genes. Currently, they have identified 20,000 plus genes. The genome is the whole things that are bundled up in these 46 chromosomes. Actually, the DNA is like an instruction book. It contains instructions for living things like you and me to develop, grow, and function. Let's understand the gene expression data using an example. Let's assume a light bulb. When we switch on it, it will emit light. Likewise, we can consider the bulb as a gene. When it comes across the gene expression controls, the chemicals that will trigger the gene, it will produce gene expressions. It can be in different levels, like the brightness. Brightness can be varied. The same time, the gene expressions also can be in different levels. So these are the data that I'm going to analyze in this research, since gene expressions are the data that better illustrate the biological process happening inside a cell. Actually, what is this cancer? Cancer is a result of an abnormal growth of cells that have the ability to invade or spread to other parts of the body. Actually, cancers occur because of the mutations happen inside the gene. The mutations happening to the genome will create cancers. All the mutations are not cancerous, but some of them are. So that's why we call cancers a genetic level disease. Endometrial cancer is the most commonest gynecological cancer that affects mainly postmenopausal women. Actually, according to the Cancer Statistics Center, there are 61,880 cases estimated to be in 2020. Why we are talking about endometrial cancer is I have applied the gene expression data of endometrial cancer to the methodology that has been proposed at the latter part of this research. Endometrium, where the endometrial cancer occurs, is a hormonal receptive organ. Ovarian hormones regulate endometrial cells' proliferation, regeneration, and functions. Every one of us have encountered cancers many times in our life, from our neighbors, friends, and etc. Cancer is not a disease just like the other normal diseases that can be treated through basic principles of medicine. If A and B having a normal disease, we can cure them by giving the same medicine. But if they have a skin cancer, although it has the same histopathological appearance, the same cure given to A will cure him. At the same time, the cure given to B, the same cure, it will worsen his situation. Why? Actually, because the cancer is a genetic level disease. That's why we need to identify subtypes in order to do personalized medicine, which will increase the efficiency of the treatment as well as reduce the toxicity of it. So to identify the subtypes, we need to identify unsupervised techniques. Since we don't have pre-identified classes, we need to use unsupervised techniques to identify the subtypes. In order to analyze gene expression data, it can be categorized into basically three parts: pre-processing, clustering, and validation. In pre-processing, first of all, we have to identify a dataset. Then we have to handle the missing values. That can be done, basically, gene expression data can be handle the missing values using global, local, and knowledge-assisted approaches. But the most important part is, since we are going to identify a hidden pattern inside this, we have to impute the missing values very carefully without spoiling the pattern. And then we have to identify the features, because actually gene expression data are high dimensional data. Since we only have few amount of patients, like 500 plus patients, we have genes, 20,000 plus genes. So that will be a high dimensional data set. So in order to identify the most important features, we have to remove the noise. So we have to use different kind of feature reduction techniques. In subtype identification using clustering, using gene expression data, most of the studies have used highly varied genes as the features. And then we have used clustering. First of all, we have assessed the clustering tendency of the dataset that we have pre-processed, and then identify the optimal number of clusters using elbow and sit-out methods, and do the clustering using hierarchical clustering with different linkages and distances, and also k-means and time clustering. After that, the validation of those clusters can be done. It can be done relative validity, internal validity, and external validity. When we are doing relative validity, we can change the number of clusters, and also we can change the measures and distances and linkages and check it. And then for the internal validity, we have used the Sirout index, Dunn index, and connectivity, which are statistical methods. And then for the external validity, we have used visual assessing method like heat maps to check whether these clusters are meaningful with the genes. Data gathering was conducted through CBio portal. In CBio portal, there were two different cancer types available related to uterus, which was uterine carcinoma and uterine corpus endometrial carcinoma, which is a type that we have used. So in there also there were 3 studies available. According to that, there were the pan cancer dataset which was updated recently. So we have used that dataset which has 529 patients with 20,472 genes. So when we are going to normalize, we have done it to eliminate the gene expressions of unaltered genes. Since we had gene expression data as well as copy number variation data, in order to eliminate these unaltered gene expressions, we have done it gene-wise. Let's assume the gene 1, the gene expression of the gene 1, and we have subtracted the mean of the unaltered genes, which are taken as copy number variation equals to 0, and then divided by the standard deviation of unaltered genes, which gives us Z score of the gene expression data, gene G1. And we did the same thing for all the genes. When handling missing values, we check the patterns of the missing values inside the data set that we have got. So inside the data set, we got 100% missing values for some genes and more than 50% missing values for some other. So when we're going to impute these kind of data, it will spoil the hidden patterns in the data. So we have eliminated all these missing value genes and taken the rest of the genes. Then in the feature selection methods, we have used 2 types of data sets, which are high coefficient of variance genes. We have selected 1,000 of high coefficient variance genes, and then hormonal genes. Since this is an endometrial cancer, we have used the domain knowledge. Endometrial cancer is occurring on the endometrium, which is the cell proliferation, functions, and regeneration is affected by the hormones. So hormonal genes will have a bigger impact on this kind of cancers. So we have used MCPI to identify hormonal genes, which are, there were 170 hormonal genes available. And from those 170 hormonal genes, we have selected 25 considering literature about endometrial cancer. Then we assess the cluster tendency using visual assessing technique as well as Hopkins statistic techniques. In Hopkins statistics technique, we check whether the data set is clusterable, whether it is giving meaningful clusters. If the data set is generated by a uniform data distribution, then it won't give good clusters. So according to both of them, we can say that both the data sets are clusterable. Then we have used clustering techniques like k-means, partitioning around medoid, as well as hierarchical clustering. In hierarchical clustering, we have used different distances and linkage measures. Optimal number of clusters have been identified using elbow and Sirout methods, for the both data sets for the hierarchical clustering, k-means and pan. When considering the internal validity of the clusters that we have identified using connectivity, Dunn index, and Sirout measures, we could see that the PAM clustering method also behaves same as the k-means, that is sensitive to the outliers, and internal validity also very low. So we have removed the PAM clustering and proceed with the k-means and hierarchical clustering. External validation was conducted for the clusters that has been identified better from the internal validation measures. So this was done visually by heat maps. This heat map was drawn for the high coefficient and variance gene data set. Using heat maps with cluster 3 and 4 identified which Spearman's distance and complete linkage were identified as meaningful clusters having good discrimination of expression levels between clusters. Heat map with 3 clusters are shown here. Heat map with 4 clusters shown here. Although we try to align the identified clusters with the clinical variables like this, like subtypes, grade, type, tumor type, likewise, but it didn't show good distinction between the clusters. Due to the high dimensionality and the heterogeneity, it was really hard to identify the most meaningful classes. But in hormonal gene dataset, we could have identified 15 clusters, and also using between sum of square and within sum of square ratio, we could have identified clusters, genes which are differentially expressed inside those clusters. For example, this gene is only highly expressed inside this cluster. So likewise, there were 15 genes has been expressed differentially in these clusters. But in here also, we couldn't align the clinical variables with these clusters. Conducted a pathway analysis for these 15 genes identified, and most of these genes are gonadotropin-releasing hormone receptor pathway genes, and also others were related to the insulin pathway. Now let's go to the discussion and conclusion of this research. Identifying better subtypes to improve the target therapeutics with significant gene was the aim of the study. Use 2 datasets with high variance and hormonal gene dataset. Use different clustering techniques and distance measures. Although the clusters are statistically meaningful and valid, it is necessary to identify meaningful clusters which was validated using heat maps visual method. K values identified through sellout and elbow methods validated using internal validation measures and then filtered considering the heat maps. The better subtype was cluster 15 identified using hormonal gene dataset, identified significant genes using between sum of square over within sum of square. Cluster 15 subtype has discriminated hormonal genes which are highly expressed in one cluster at a time. Significant genes in cluster 15 are connected with gonadotropin releasing hormone pathway and insulin pathway. High dimensionality of the data is a huge limitation that came across when interpreting results and identifying the important features. Difficulty facing validating the clustering result is a limitation, as although the clusters can be identified and validated through statistical methods, meaningfulness of the clusters are difficult to identify. Future works can be use deep learning methods in order to select features like autoencoders to capture the underlying genetic patterns and select the most meaningful features. And also integrate all the types of genomic data like mutation data, copy number variation data, and expression data to identify the clusters. And then also identify better methods to find gene signatures in clusters. That's the end of this presentation. Thank you for listening.
Very much, Amali. So, actually, in your title, you say analysis of expression data using unsupervised techniques. So, actually, what's your major contribution on our team this work? Can you please turn on your camera and answer?
Madam, can you hear me? My camera.
Can you increase the voice? We can't hear you.
Can you hear me now, madam?
Yes, now we can.
My camera is not working, I think.
Yeah.
Yes. I think in this research, madam, I have proposed a methodology to analyze gene expression data like a pipeline. And also I have compared different clustering methods, which I have identified from the literature that has been used to analyze these subtypes previously, like there was a study which was done for breast cancers, which have identified 6 subclasses. They also have, and also there was a study for the endometrial cancer. They have identified 4 subclasses, including the expression data. And they also, most of the cancer-related subtype identification studies have used these clusters. So I analyzed them and proposed a pipeline, as well as the dataset that I have been used was taken as a statistical dataset as well as a domain knowledge-based dataset. So from that domain knowledge-based dataset, I have identified good clusters with gene signatures. At the end of this research, what I have identified was, since we don't have a ground truth for the cancers actually, so we don't have a method to validate the methods, the clusters that we got, but anyway, although they are internally validated means the main thing that we want from clusters is to be compact between the data points inside that cluster and to be separate from the other clusters. Although we can measure it from internal statistical validations, we don't know whether the ground truth is we got that ground truth. So in order to maintain the meaningfulness of those clusters, we have to identify the hidden patterns of the data, the genomic data set. So in order to do that, we have to identify better features because of the data set that I have got from hormonal genes that is related to endometrial cancer, that is taken from the domain knowledge. So that gives better clusters, but the 1,000 genes that I have taken as the highly varied genes didn't give much better clusters. So for the follow-up work, I'm trying to try to build a autoencoder or neural network to capture the biological domain knowledge from the data.
I think in your future work, it's better if you could, choose a different data set which has the ground truth. I don't know whether it's available. However, if you can choose a data set which is used by some related work also, you can at least compare at the end. So that's my suggestion. Okay, thank you very much for answering my question. Are there any questions from the audience? Seems that there are no questions, so we can proceed to the next. Thank you very much, Amali.
Thank you.
Presentation is for the paper ID 124. The title of the paper is Hub Genes Identification in Brain Cancer. Expression data to so this will be presented by Senadira, SPBM Senadira. So he's a PhD student at University of Columbus School of Computing. He had my PhD internship at EMBL EBI UK, and he have completed his master's reading regarding bioinformatics, and his research interests are cancer genetics, clustering, and network analysis. So let's invite Senadira to do his presentation.
Hello, I'm Bhakya Senadira. My supervisor is Dr. A.R. Veerasinghe. Our research topic is hub gene identification in brain cancer with gene expression data. This is the overview of our presentation. I'll start from the introduction, problem statement, objectives, methodology, results, discussion, and conclusion. Cancer is a main health issue in worldwide. Brain tumors, brain cancers are the 8th most common cancer overall among persons aged more than 40. Brain tumors are the 5th leading cause of cancer-related deaths in males aged between 40 to 59. The 5 years relative overall survival rate for adults over the age of 40 diagnosed with a brain tumor is only 21%, which is significantly lower than the other cancer types such as colon cancer and breast cancer. So, analyzing about the brain cancer is a significant research aspect. So, we are trying to understand the gene expression level variation in brain tumors. So, I'll explain what is gene expression. So, DNA will convert to the RNA and it will convert to the protein which will do all the cellular tasks. So, if you analyze the RNA level or the RNA count, we can get an idea how the protein level work in our cell. So whether the function is over represent or under represent, we can identify from gene expression level analysis. So in here we are counting the RNA numbers. So by counting these RNA, we can identify what are the genes expressed in our cell for in a particular state. In order to understand this, we are using network analysis. In human cell, proteins interact with each other in order to perform a task. So proteins work as a network in order to do a function in our body. So these all functions get together and created a human interactome, which currently it has 11,999 proteins, and these interactions are published or were studied in biological context, and these are published as interactions. We call them links for these proteins. And there are 74,771 interactions in our human interactome currently. So, we are trying to analyze this network and identify density-based subnetworks for these brain tumors and also the complete graphs in brain tumors. The problem statement of our research: the knowledge gap between critical genes in human nervous system cancer subtype called PCPG, which has gene expression level alteration. The objectives of our research is as follows. Main objective is to identify protein interaction networks and hub genes in the brain cancer, PCPG, and the sub-objectives are to identify proteins which are highly connected and hub genes of highly connected gene networks. We have used the following methodology. We have downloaded the data and filtered them, and then we have do the gene filtering, and we have generated the networks, and we have identified the hub gene, the main significant genes in those networks, and we have validated the results. The data that we have used for this analysis as follows. We used the CBAR Portal 2018 June version for download the PCPG data, and it has 20,987 genes and 184 donors. For the gene filtering, we have used coefficient covariance. In order to filter these genes, we have calculated their coefficient covariance, and we have identified in 0.5 there is a significant variation which has an elbow as explained in this graph. And from that we have come to a conclusion that less than 0.5, these are the housekeeping genes in order to do the normal cellular functions. And they are not significantly involved in cancer, so we have eliminated those genes which has less than 0.5 coefficient covariance. On the other hand, more than 5% is considered as error in the gene expression sequencing. So after the filtration, we got 184 donors, and also we got 4,191 genes. From that genes, we have developed a network by using human interactome. The human interactome has all the genes and their interactions, and from that we have mapped our PCPG genes and extract the network. So this network has 3,924 genes, and their interactions are 40,450. And it has 341 components, and the clustering coefficient is very low as 0.124. Then we have used M-code algorithm, which will identify the highly dense region in the interactome, and we got two main clusters, M-code clusters, which are represent in our PCPG dataset. And this M-code clusters have 38 score for the first one, and the second one has more than 10 score levels, and they have 43 genes and 800 edges, and the second one has 24 genes and 118 edges. Then we have analyzed with the cluster 1 algorithm also the PCPG interactome, and we got only one highly dense region from cluster 1 algorithm, and this is a kind of expansion of M-code result, which has the M-code cluster 1. And then we have merged these three clusters, gene clusters, together, and identify that these two of these clusters are mapping together, as you can see in the right side. And there is another cluster which is totally different, M-code cluster, totally different from other cluster 1 result. In the next step, we have identified the hub genes of each network that we have identified from M-code and cluster 1. The M-code cluster 1 and cluster 1 result has very similar hub genes, and it represents the RSP ribosomal gene group, and it is involved in tumors as indicated in omic data. And then for the M-code cluster 2, we have identified several oncogenic genes such as LIMA1, CDK16, and ZNF638, and NADK. These are highly involved in tumor progression. Then, for the hub gene identification, we have used the merge gene network also that we have got from merging all the three networks arrived from M-code and cluster 1. From that, we have identified this ribosomal group, gene group, which is highly involved in cancers for gene regulation. In the PCPG protein complex, we have identified a complete graph. It means every gene of the network connected to all the other genes. So in this scenario, this is very significant in biological context, since they can be a protein complex. As we discussed earlier, we have identified a ribosomal protein complex which involves in tumor progression, and also they are maintaining the cell proliferation. And then the ribosomal protein S4 are playing a role in tumor syndrome also. So the findings are critical, and they need to be further validated with biological evidence. The conclusion of our research is ENCODE and cluster 1 able to find highly density regions in the large interactome compared to the MCL, and FAG and EC algorithms. So we have used several other algorithms for this analysis. However, they could not generate any results. And we have identified two significant genes, RSP4X and RPL73A, which involve in PCPG cancers, and three important protein-protein interaction networks identified, which we can use as a marker for PCPG cancer detection. That means these protein set can be used as a identification gene panel for PCPG cancers. And also we have identified complete graph of ribosomal protein S4 genes as a hub genes. So these are the conclusion of our research, and these things should be validated with biological evidence in a biological lab procedure. Thank you.
Okay. Okay, thank you very much. I have few questions to ask from you. Can you please turn on your camera? Findings. So, so is your contribution majorly on biological insights on the application?
If I see it from the camera, my signal is a little bit low. Yeah.
Yeah. So your contribution, is it mainly on the biological insight?
More about the biological insight. So I think you can hear me, right?
Yes, you can.
Yeah. It's mainly about but however I compared several algorithms such as MCL and clustering.
So didn't you find any related work have they ever not tried to identify genes in
algorithms give any results. Can you hear me?
There are some problems in the communication, I think. But I asked was actually
no, actually I didn't find any literature regarding brain tumors and so on. However, there are this kind of protein network analysis have been done.
Haven't been done yet. I find some.
Sorry, I didn't hear you properly.
Okay, I could find one publication. This is for you, future. So identification of hub genes and pathways in one of the brain cancer types, glioblastoma, by bioinformatics analysis. So you can compare your work with that. I think that would help you to evaluate your results.
Yes.
Okay.
Yes, I use other analysis. I use that. This is the data set is the popular data set for brain tumors, and this is kind of a sub type of another brain cancer type. So I use this for this one.
So I think you would be also able to find out those genes they have used, isn't it? They have identified as subgenes because this is subtype.
Yes.
So, all right. I think you are continuing your work. I think this is part of your PhD.
There are some similarities. Yeah.
Are there any questions from the audience?
Yes, madam.
If you have any questions, you can ask from her now. Okay, it seems that there are no other questions. So I'd like to thank you all the authors, who are say Nadira and Kalana and Amali. for presenting their research work in this bioinformatics session. So, and also the people who helped me sitting on back, also I'd like to help for the cooperation given, and also for the audience for their questions and enthusiasm. Thank you very much.